So we are continuing with the spirit of the topic of membrane potential of cells. And this will be us wrapping up the subject in preparation of the action potential. And we're going to discuss the last contributor of our membrane potential of cells, the Donen potential. And we're going to go over minimal requirement question from the Department of Biophysics and Cell Biology from the University of Debrecen. And I find it really important to not only remember those minimals, but to understand them, because even if you're not actually sitting down on part A of the exam and you're exempt from the minimals, these minimals that we're going to go through are represented as open essay questions very often. And we're going to go through some question solving after we go through the minimals, and you will have a chance of assessing how well you understand these past two lectures. So let's get started. And I want to discuss the Donen potential. And the Donen potential is a small contributor to our entire membrane potential. And what we said is that our membrane potential is due to uneven distribution of ions across the membrane. That means that if I have around 3,000 positive charges here, and on the outside I have 4,000 positive charges here, that affects the affinity of these molecules, or more simply put, that affects the desire of these positive charges to go in. That affects that desire because they want to go in because there's a low concentration and they really want to go in. So this is slightly more negative, whereas the environment is slightly more positive. Now the Donen potential says, okay, it's nice and it's good to have uneven distribution of ions that generates a slightly more positive or a slightly more negative charge inside the cell. But also we have proteins inside the cells that are very, very big molecules that are negatively charged. We have negatively charged proteins, and they're really, really big molecules that are negatively charged. And these big molecules are negatively charged and can't really escape the membrane. They cannot leave. They're staying inside the cell, and just being there and being negative, contributes to the membrane potential. And why is that? When I'm saying membrane potential, we should know by now, that I'm talking about a relatively negative charge inside the cell compared to the extracellular environment. And if in the extracellular environment I have more positive charges than I have inside the cell, then the cell is going to be negative compared to its environment. What I'm doing here is actually adding negative charges. I'm adding negative charges inside the cells that cannot escape. So now these are effectively adding to the desire of these molecules, to go in. Because before, these molecules wanted to go in, yes, but now there's even more negative charge, so they even want to go in more because the cell is even more negatively charged compared to its environment. So the Donen potential, and this doesn't really necessarily mean big proteins, but this is just big macromolecules that can't escape. It's just easy for me to think about proteins. And if you're asked in an essay question and you say negatively charged proteins, you will be correct. So whatever way you choose to remember it, but we can say the presence of macromolecules negatively charged that cannot escape, add to the membrane potential, or add to the membrane negative potential. Now let's try and understand, is it a thermodynamic equilibrium idea or not? Well, if we're looking at these molecules, they can't go out, and they can't, and none of these molecules, if we have these molecules outside, none of them can go in. So there's no net transport of material. So this would necessarily mean that this is a thermodynamic equilibrium. This is a thermodynamic equilibrium. Just because there's no net transport of this big macromolecules across the cell membrane. So this is a Donen potential. There's really not a very massive topic. You just need to know, what does it mean? It's the final contributor of the three contributors that we have to our membrane potential. This is a good time to mention those. Our contributors, or the factors to having to my resting membrane potential. First and foremost, the highest impact is the diffusion potential. And by diffusion potential, I mean the Goldman-Hallstein-Cotes equation. The second one is the, let's just say, the pumps. We have sodium-potassium pumps that help take out 3 positives and put in 2 positives, and by that they are electrogenic and they help us maintain the negative membrane potential. And also we have the Donen potential, to a small extent, the Donen potential. And from research that I have read, it accounts to roughly anywhere between 12 to 20 negative millivolts out of the, let's say, negative 70. So these are the major contributors. These are the, or rather, this is the major contributors, and these two are the smaller contributors to the negative membrane potential. Perfect. And now all we have left is to brush off on these minimal requirement questions that, if we look at them, they may seem kind of daunting. And in effect, we can break them up to make them to make them more simple, not break them up to make them more complicated. Then you would probably not be watching these videos. So let's take a look at minimal 213. And again, this is brought to us by the Department of Biophysics and Cell Biology. So, what can the explanation for the decreased lateral mobility of a membrane protein? Well, if we think about it, when we have membrane proteins that we said, if this is the bilipid layer and we have proteins here, we already said that these proteins can move around. And the question is, what is going to decrease their ability to move around or to restrict them? First of all, anchoring by a cytoskeleton really means, and I don't really imagine that you need to remember that in specific because it didn't go through this with us, but we have specific peripheral proteins. If this is the extracellular, let's say this is the extracellular, and this is the intracellular, we have peripheral proteins that are not spanning the entire length, they're just on the outside, and there can actually be a process in which they are anchored via some sort of element here to the bilayer, and they cannot move. And this is really anchoring. And when a protein is anchored, it cannot move anywhere. And what do we have? Association of the protein with other membrane proteins. And we already said that we can have, let's just say, this is my plasma membrane, and this is my sphere. And I have a process that needs to take place here. And for this process, those lipid rafts, my lipid rafts herded up all of my proteins. I need, let's just say I need a green protein, and I need a blue protein, and I also need an orange protein. All of them need to be in the vicinity of one another for this process to take place here. And if they're interacting right now, they can't really move away from one another. So this is basically the association of these proteins, association of proteins with other proteins. Let's get this going. Aggression of the protein in question, this is basically all that it means. If the protein is being aggravated, then its mobility and its functions are being subjected to change. Localization of the protein in question at the membrane contact surfaces. And again, this is not really something that can be interpreted. It's just that proteins move around. And if they move around, and we're talking about a specific protein that has a contact surface at a given point, and that contact surface limits its motion, then it's not going to be able to, or its lateral mobility is going to decrease. Obviously, decrease in membrane fluidity, I think this should be right on top here, because this is a major contributor. If my membrane is in a gel state, and everything is really packed together, it's going to be considerably harder moving around through it. And this makes sense, or at least it should. Hopefully it does. And the protein cannot cross the border of lipid domains. And what do I mean by that? Let's just say, and we have the detergent insoluble glycolipid, the lipid rafts, whatever it is. And these are my lipid rafts. And let's just say this is my bilipid layer. And these detergents insoluble glycolipid microdomains, oh, that's what it is. Let's just say there's a protein here that wants to move, but it can't move through it. This is a lipid domain. It can't really move right through this domain. So this is proteins cannot cross the border of lipid domains. This is just an example. There are other examples, but I'm just trying to give you some sort of intuition so you can understand these things in case you're asked about them. And I believe that you are less likely to list this as an essay question, but it's just nice to understand it. Minimal 219. What are the properties of facilitated or carrier-mediated diffusion? And basically what we're asking is, let's just say I have a channel here. What is the property of having this channel transport ions inside my membrane? Inside or outside doesn't really matter. So I have my ions and they're transported in. First of all, they're specific. They specifically bind to their transporting molecules, meaning that if this is a potassium channel, only potassium is going to go through. And we're going to learn about it later, how nicely selective it is, and it's a pretty interesting mechanism. And it also says that, hey, if this is a sodium channel, only sodium would go through. So this is, they're selective. So this is the first real thing, and selective, specific, really the same idea. And it is inhibitable by a specific inhibitor. What this really means is that if I have this little guy here that for some reason resembles a potassium to a great extent, but instead of coming over here, it can just block it. It can just block it entirely, and when it blocks it, no potassium can go through. This is an inhibitory event. So these channels can be inhibited, like any form of you can say, facilitated diffusion. Inhibited by specific inhibitors, we went through this. The flux of transport can be saturated over a given concentration. Basically, this is a really funny way of saying, hey guys, if I have, let's just say I have 3 channels. I'll probably have more in my plasma membrane, but let's just say I have 3 channels, and each channel can transport, let's just say, 10 of these ions per second. 10 of these ions per second. 10 per second, 10 per second, 10 per second. Let's just say that there's this crazy thing going on and I need all these channels open and all these channels are going to work. I am not going to be able to get more than 30 ions per second. I'm not going to be able. And this is what we mean by it's saturated. It cannot undertake more ions. It's to the limit. And that's what we mean by can be saturated. Very good. So we're slowly making head. We're slowly making head. Do I have some more on this side? Yes, I do. All right. Very good. So we already went through this minimal. That's the different contributors. And we mentioned the diffusion potential, which is number 1, which is the Goldman-Hodgkin-Cutts equation. And these 2 are the minor contributors. And what I myself don't really like very much about this minimal requirement question is the use of a term that they never use with us. And this basically means resting. This basically means resting. So list the factors contributing to the maintenance of resting membrane potential. And I for the most part, like to keep to the terminology that I use. I don't like to switch around because I find it confusing. So this minimal, what is this about? Describe the relationship between the net flux of major permeating ion in the case of resting. Oh yeah, this one's a favorite. Well, basically what we said is that this Goldman-Hodgkin-Cutts equation gives us what is going on inside a cellular environment. And inside a cellular environment, and I've been drawing circles a lot lately, but in this cellular environment, we said that we have, let's just say, an uneven distribution of ions if we compare to the outside environment. So this is going to be partially negative. And whatever happens, I need to keep this partially negative charge. So let's just say I'm taking in a potassium positive charge. That means I need to eject some sort of positive charge, or maybe take in a chlorine. Or let's just say I am losing a chlorine, because I haven't been talking about chlorine much, so I'm just going to do that now. If I'm losing a chlorine, I'm losing a negative charge. So I can either, let's just say I can maybe lose a sodium. and that is going to balance it out. And you can pretty much imagine the only thing we have left is to put in sodium, and that way I can maybe eject potassium. That's really the basic ideas that I want to convey. And what this means is that the total flux of all these guys, the total intake and outtake and all the motion needs to cancel out as far as charge. There needs to be some sort of canceling out. So the net flux of all these guys need to be zero. That means if I take one in, I need to put one out. So the net flux is zero. Different material is going through, yes. So there isn't well, you can say that we have an uneven distribution of material, but as far as charge, the net flux needs to be zero. Or you can think of it, for each ion, one goes in, one has to go out. So the sum of them needs to be zero. I know that I sometimes tend to repeat myself often, but you can always fast forward if you feel that you've gotten the point. And now what I want to do is I want to present you with some questions, and I want you to take the time and freeze the video now. Freeze it now so you can work on it. So freeze the video. Do it. Seriously. All right. So to whom of you that have been working on these questions, let's get started. We have a fill in the blanks, which is a classic. Usually I have a word bank, but I'm not going to make it easy on you. Let's see, a primary active mechanism, which could be, and this is how I solve these things. I just write pump. A primary active mechanism, transport ions from the point of something concentration to something concentration. And this is not a too difficult question. If I'm using energy, then I'm going against the gradient. If I'm going against the gradient, I'm going from low concentration to high concentration. Because if I'm not using energy, I'm going from high to low. Ions cannot pass the cellular membrane because of their, and we mentioned what kind of molecules cannot pass. Well, we said that polar molecules, or very big molecules, or charged molecules, and ions. Potassium is not polar. It's not very big, but it's charged. So I would say because of their charge. This is what I would say, because of their charge. Let me just undo all this mess. All right. So phospholipids are a type of molecule, and this was actually, I'm not sure if it's in the minimals, but it's well represented in the lecture slides, and a molecule that has a hydrophilic and a hydrophobic domain is called amphihepatic. Amphihepatic. Perfect. Below the transition temperature of the membrane, the cholesterol would usually something viscosity. So we're assuming it's going to either increase or decrease viscosity, and I'm going to do it just now as I would in my exam. I would say, okay, I have, and I do drawings when I'm in my exam. Sometimes I draw smileys. Don't do that. They don't like that. So this is my transition temperature. This is my liquid phase, and this is my gel phase. Perfect. So below the transition temperature of the membrane, that means over here. So when we're in a gel phase, the cholesterol would usually, what would it do? Well, the cholesterol would want to increase fluidity. It would want to increase fluidity because it's in the gel phase. It would like to counterbalance this. And in order to increase fluidity, we need to decrease viscosity. And this is what I would do real quick in my mind when I'm doing an exam. So it would decrease the viscosity. It usually decrease viscosity. Carrier ionophores are a form of something transport. What type of transport do we know? Well, the main two types of transport are either active or non-active, passive. So we have passive and active. And I know that ionophores carry ions from high concentration to low concentration. And even if I only remember that, that would be enough, because high concentration to low concentration is always passive. Very good. So true or false? Let's see what we have here. And again, this is a good time to pause it if you want to work on this particular part. So let's get cracking. The part of the phospholipid Phospholipid facing the extracellular environment has a hydrophilic affinity. And that really means that if this is my bilipid layer, and obviously I'm drawing it very, very shitty, but if this is the bilipid layer, they're asking about this part. So the part of the phospholipid facing the extracellular environment has a hydrophilic affinity. And if I'm not really sure, I know from chemistry that fatty acid chains are hydrophobic. So the inside is going to be hydrophobic, so the outside is going to be hydrophilic. True. Let me just wipe out all this artistic things going on. True. Very good. A non-zero net flux of ions, and these are just the terminologies that they use. A non-zero net flux of ions across the membrane describes the situation of thermodynamic equilibrium. All right, let's see. In thermodynamic equilibrium, let's say that we have a thermodynamic equilibrium. If I have some sort of transport, for each one going in, there's one going out, so there's no net transport of material. So there is a net zero flux. So what I do in exams is that I would just cross this over, and read the sentence without it. If the sentence makes sense, if after I fixed it, I would mark a false. A zero net flux of ions across the membrane describes the situation of thermodynamic equilibrium. This sounds good to me. So this will be false because I had to fix it. I had to fix it for it to sound good. Ionophores and facilitated diffusion mechanisms both transport ions with the chemical gradient. And this is just interchangeable terminology. With the chemical gradient just means from high concentration towards low concentration. And I know ionophores do that because we just solved a question about it. It's passive. Ionophores and facilitate diffusion mechanisms both transport ions with the chemical gradient. When you think about it, the only form of transport that transports it against the chemical gradient is active. And we're not really talking about active because facilitated is a form of passive. So I would say this is true. Both ionophores and facilitated diffusion are with the chemical gradient. The Donen potential addresses the permeability of ion species. And obviously this is incorrect because we already mentioned that the Donen potential addresses molecules that can't really move around. They can't really get out or get in. And what I would do is I would just fix this, and instead of this, I would write the Goldman-Hodgkin-Cutts equation, and then I'd read it out and see if it makes sense. The Goldman-Hodgkin-Cutts equation, or Goldman-Hodgkin-Cutts potential, addresses the permeability of ion species. Yes, this seems okay to me, and being that I had to fix this, I'm going to go ahead and mark a false here. Very nice. Let's keep going. At electrochemical equilibrium, there is a net transport of ions across a semi-permeable membrane. Let's see what this is, the electrochemical. I'm not going to draw all of it, but I'm just going to pass the message. For electrochemical ingredient, I have an area that is slightly more negative and an area that has, let's say, more concentration of potassium, and here less concentration of potassium. You know what? I'm just going to draw it. Because it's going to be nicer to see it this way. Let's assume these blue dots are all positively charged. So for each blue dot going here, because it's with the chemical gradient, because there's low concentration there, one blue dot goes in here because there's a very negative charge. So, and this is electrochemical equilibrium, and there's really no net transport. Otherwise, it wouldn't be called an equilibrium. So if I'm trying to fix this and then answer, I'll just say, no, there is no net charge, net transport. Let's see. At electrochemical equilibrium, there is no net transport of ions across a semi-permeable membrane. Yes, I agree with this. So I would mark a false for this one. Ionophores have antibiotic properties, and this was actually a question from a very recent exam in 2011 in the second self-control, and this is true. And if you're wondering what are antibiotic properties, well, you can think about the layer tries to block ions from going in. It tries to block ions from going in, and it has some mechanisms to get them in. So it has a very ordered protocol for them to go in. And for these ionophores to come in, grab these ions and just move them in, it just interferes It interferes with the plan of the plasma membrane. So being that it's interfering, it has some sort of anti-biological property. A secondary active transport mechanism uses ATP. When we think about it, we already mentioned that what uses ATP directly would be the pump, and the pump is going to generate a gradient, and the secondary active mechanism is going to use that gradient. But when we're saying a secondary active transport mechanism uses ATP, some people may say, yeah, well, it uses the ATP from the pump. The answer is no, it does not. It does not use the ATP from the pump. It uses the chemical gradient. The only thing that uses ATP is a molecule that hydrolyzes it. And the secondary active transport does not hydrolyze it. So this would be false. Or I can say a secondary active transport mechanism does not use ATP. So that would be true. I really, really hope you found this helpful. I only added this whole portion of problem solving because I find that when I have a very complicated subject or a subject that isn't intuitive like NMR, it is an easy way for me to attack it from the other end, to attack it from what are they going to ask me? Am I ready for those type of questions? So hopefully you found this helpful, and let's move on to action potential.